Recording's in progress. Okay.
Right. All right, off we go. So, so today I'll be telling you about how fish feel, the sixth sense, and what it can teach us. Oh, why is that happening? Oh, it's zooming. That's weird. I didn't anticipate that. I click
And
when I
it's,
and it zooms instead of going to the next slide,
do you have the,
and the arrows are moving it around.
wait, do you have like the buttons on the sides for
Oh, I don't even
going?
see them. There's no
Um,
button.
is this, this is the snorkel link for the
There,
presentation?
now I see it. I hid some set of controls that I shouldn't have hid, apparently.
Okay, got it. So, all right.
So
So why
now
don't
I
you,
have it.
yeah, why don't you reload? Yeah, go
Yeah,
back and reload.
there.
So, okay. So maybe just keep this to this way. Is
Yeah,
that?
because when I was hiding, do you see this stuff up here?
Yeah, yeah, I see that. And so when you're
When I
hiding,
hide that, then it
then
doesn't, I
the
lose
arrow
these
also gets
buttons.
hidden.
Yeah.
Okay. I'm glad we're
And
doing
then
this.
when I tap it, it zooms instead of
Yeah, got it.
so I'll just keep that in mind. Okay.
And then when you go to the next slide,
Takes a
okay, and then
moment to
this
load.
one I don't see the controls on, so this one seems like maximized appropriately.
Hmm.
Yeah, I guess it was just that one, that first slide there. But okay, I'm glad to know that. I'm glad we're practicing that. Okay.
Okay, good, good. So that's the second fish. That's the first fish. This is a flat image. So this is super easy. Um, I can zoom in and be like, there's no eye. Where is its eye? Um,
Does it actually not have even any infrastructure for
it
like
still has the orbital
receptors?
bones, but the eye itself, like the lens degenerates and the I believe the retina
Okay.
as well. And they actually develop a more complex lateral line with more and larger neuromasts. Um, so even though it's these same species, these two, they can breed fine, they produce viable offspring. Um, but they look dramatically different, and it's basically all epigenetic. I'm not going to go into detail about that, but I'll just say that there are non-genetic changes that make these fish able to navigate in the dark, and they use their lateral line system. So, you know, like that.
Awesome, awesome.
Yeah, yeah, yeah. And then I also added this little friend, an actual zebrafish, just to be like, this is what we study, right?
That's
So
pretty cool. That it
yeah, I
looks
was
like,
glad I was able to find this.
yeah, it's like surface photogrammetry or something like that,
Yeah, I don't know how they did it exactly, but it's pretty sweet. Like it looks
yeah,
great.
because it's like the thing they probably yeah, nice.
I mean, it's a little angular, you can see like the sort of polygons or whatever,
It
but
almost looks like someone like carved out the mouth, but like maybe the rest of it is actually from real scan.
maybe, yeah. I found images from real scans, like from micro CT. There were some pretty
Yeah.
decent ones, but they look more, they don't look like a zebrafish. They just look like some random fish. They're just gray, like they don't have the stripes and they look a little more, I don't know, gnarly, I guess, because they're real, so they've been through the ringer. So this looked a little smoother, but a little prettier, I guess. So
Nice.
just to mention, this is the fish we study. They are native to India and Bangladesh and Pakistan, and these fish are very commonly used in laboratories because we know a lot about them. We know how to study them. We know how to manipulate their genes. We know we can breed them and make lots of them. So all of these tools make them great as a what we call a model system, meaning an organism we can study to learn about how things work in other systems, other organisms, yada yada yada,
Nice,
etc.
nice.
Um, and then let's see, these are the toughies, right? So this one I zoomed out a little bit,
Yeah.
pretty smooth now,
Oh yeah, that's pretty smooth for me here.
right?
Yeah, yeah, yeah. Yeah.
Okay. I
I
may
might
zoom in on a
like,
neuromass, which gets a little choppy, but I'll just do that like for towards the end just once, and then
yeah.
I won't do a lot with the zoomed in view. Um, but I may like just sort of center a neuromast and then zoom way in to be like, this is one sensory organ. something like
So
that.
yeah, totally. Actually, in the future, like not
Mm,
within the next 2 months, we're going to be using a dynamic loading method for volumetric. So sort of like the Google Maps thing for 2D, except in 3D, so that
cool.
so that it won't have to have all that other stuff in memory, and it should be a lot smoother.
Awesome. All right, good. I changed the order a little bit here. I switched to having the neuromast next, saying like these are the sensory cells within those little sensory organs. These are called hair cells. And together they make up a neuromast. Neuromast is the sensory organ of the lateral line. And if we rotate this, and this is a little less smooth, I zoomed it out a bit, but the problem is this gets really small. You know what I mean? Like if I
Yeah.
zoom out much more than I have,
Yeah.
oops, I just screwed something up. I tried to open something I didn't mean to. But if I rotate this, like, if I zoom out, it's going to be a little smoother, obviously, but
So
it's just tiny, like,
yeah, so actually, I should say, even
yeah,
though I know that your experience is choppy, but like Zoom is normally a little bit choppy, and it actually doesn't look super different from normal Zoom choppiness. It looks a little bit more choppy, but I think like people are
it's not
comfortable.
terrible. Okay.
Yeah, like I'm watching this, and this just seems like normal Zoom latency. So
Okay. All right, great.
yeah.
Good. And then we're into the models. This is what a single hair cell looks like. And the way it works is force exerted on this top part, which is called the hair bundle, is translated or transduced into a signal coming from the base of the cell down here and going to these neurons. And that sends a signal to the brain that you're hearing something, or that the fish is sensing something. Now, why are we interested in these cells? Well, it turns out the same exact kind of cells are present in the human cochlea. That's the inner ear where hearing happens. So we are very interested in understanding how these cells work. And not only that, but in fish, these cells can naturally regrow, whereas in humans and other mammals like us, those cells don't regrow during the life of the animal. So that means that if you lose these cells, you lose your hearing. Fish don't have that problem when we're trying to understand how can we regrow those cells in the human ear. Yada yada yada, something like that.
Nice. I just you might want to either remind people every now and then that they can move things around, or you might want to move things around a little bit every now and then, because just
Mhm.
uh, just so that it's clear that you can.
Oh yeah. I was just
Oh, okay, yeah, yeah. You're
literally,
just like blowing through it. Yeah, no problem.
yeah,
Cool.
just blowing through it because I know we're low on
Yeah,
time.
yeah,
I
totally,
just didn't want
totally,
to like
totally.
spend a lot of time just spinning
Yeah, yeah,
things.
yeah,
But
yeah, yeah.
um,
Brian Boitano.
what would Brian Boitano do?
Did somebody say
um,
my name?
what?
Hi, boys. Did somebody say my name?
Bye. I still use that routinely. Like when I leave a room, I'll be like bye, and I don't think anybody knows that I'm referring to Brian Boitano exactly.
Brian Boitano. What an
What
incredible
would Brian
honor.
Boitano do? Huh?
Brian Boitano, what an incredible honor.
All
Oh,
right.
shit.
Anyway, it
He's
seems
come
to be
to kill
working
you
fairly
because
well.
you're
Huh?
Jewish. He's come to kill you because you're Jewish, Hal. Oh,
Oh boy, that
I love
show.
I love the fact that we're the same age, and specifically this age.
You're right, exactly, because we get the same ridiculous, unnecessary references. Really critical. Um, so that's that. I mean, I think it seems like it's moving pretty well, and
Yeah, that looks great. That looks great. I'm glad we did this test, and yeah, we you can check out.
find out of this one, go into the other Zoom.
This is like the right one.
Oh,
Like
this
this
is the current. Okay,
this is the correct one. Yeah,
gotcha.
yeah,
Okay,
yeah, this is gonna be the right one.
I didn't look
So,
at the actual room numbers, so I didn't know if this was like. All right, so this is where people will presumably be joining at some point.
yes, exactly.
Great.
So, all right. Okay, so yeah, you can turn off your video. I'll check out for a while.
All right, sounds good.
All right. Bye.
Bye.
Hi, everyone. Thanks for joining. We're just going to wait another minute before I start to introduce Dr. Aaron Steiner. All right. Um, I'll keep on admitting people, but thank you all for logging in precisely at 12 or a little bit before. Um, Really happy to welcome you to our first in a series of interactive science lectures by experts in different fields, ranging from astronomy to paleontology. And today we're really lucky. I'm very happy to introduce you to Dr. Aaron Steiner. He is a neuroscientist and developmental biologist, and he leads a research lab trying to uncover how hearing works. It's a sensation you don't really think about most of the time, but it's pretty cool how it works in your brain. Um, Dr. Steiner is also the chair of the Department of Biology at Pace University. Um, and I should remind you before we start on several of the slides, there's going to be QR codes in the bottom right, and you can actually use your mobile devices to scan these and interact with the same three-dimensional visuals that Dr. Steiner is showing you there. So you can put your mobile devices to a good use. Um, Kids, it's one legitimate ask of your parents on a Sunday morning. So, without further ado, Dr. Steiner, do you want to share your slides and say hi?
I absolutely do. Hello. Yes. Hi, everybody. Welcome. Hopefully everybody can see what I'm seeing. Is everybody seeing an introductory slide that says interactive science? Yeah, it's working. Okay, great. Let me hide my controls. Okay, wonderful. So, I'm going to ask you if you have any questions during the talk. Feel free to interrupt me. I'm very used to it. I teach at a university and I get interrupted constantly, and I kind of like it. So, if you have questions, you can ask me. If you can put them in the chat, that's another option. And Dr. Fisher will, tell me about them at the end of the talk. I'm going to talk for about 15 to 20 minutes, and then I'll take questions after that. So I'm excited to be here. I want to thank Jonathan Fisher again for inviting me to do this, and the Snorkle app has made it really, really cool. I think you're going to enjoy all the visuals we're going to look at. And again, for anybody joining late, there are these QR codes down here in the corner. So any of the images that we're looking at on the screen, you can actually use your phone, or as Dr. Fisher already mentioned, you can pull up those images in a browser using the QR codes, and you can manipulate the images yourself. You can zoom in, you can move them around. So it's pretty cool. Okay, so today I'll be telling you about how fish feel, the sixth sense, and what it can teach us. So hopefully there'll be some new information in here for many of you. And let me just shrink this down so I can see what I'm doing. Okay. So let's get started. Um, I think most of you will be familiar with a fish. This is a fish, right? It's a perch. And what I want to point out to you about this fish, something you may have noticed in the past, and look at this, I can rotate it and move it right to the center. Um, so has anybody ever noticed this line along the side of a fish? Does anybody know? I'm actually curious if anybody knows what this is called, this line that runs down the side of a fish. You may have seen it in the aquarium. You may have seen it at a grocery store at the fish counter. Anybody know? We may have some ringers in the audience, but it's called the lateral line. Um, and so all fish actually have a lateral line system, and that lateral line system is a sensory system. So they use this line to detect a variety of different things in the water around them. So one thing they can detect is their movement, their own movement, how much they're accelerating and how fast they're going, which is very helpful if you're moving through a dark environment, you're moving through the water, it's kind of dark, you can't see everything that's going on. You can feel how fast you're going and your acceleration. So that's one thing they use it for. Another thing they use the system for is to feel predators and prey coming. So they can actually basically feel things at a distance using this lateral line system. So they can even feel a predator coming from behind them. They can sense prey at a distance and go after it, even in total darkness. So this is one really cool thing that fish can do that we can't. In fact, there are some species of fish. Let me see if I can just go to the next slide here. So there are some species of fish that use this lateral line to get around almost exclusively. So this fish is called Astyanax mexicanus, which is the Mexican cave fish or cave tetra. You can actually buy them in a pet store. In many pet stores, you can find these fish. They're actually about an inch long. They're much smaller, maybe 2 inches long, smaller than what you're seeing here. Um, so what's interesting is that the two fish that you're seeing here are actually the same species of fish. They can interbreed, they can make, offspring together that are totally normal. They're the same species, but they look dramatically different. And that's because these fish with the eyes up here live at the surface of the water. They live in rivers and streams, and they use light to navigate, just like we do. But these fish are a population of the same species that lives in a cave where it's completely dark all the time. And they have actually evolved to lose their eyes, essentially. They don't need them. So it would be sort of unfavorable. There's no benefit to growing eyes, developing your eyes if you don't need them. So they don't develop eyes, or they develop them and they kind of degenerate. And what they do instead is they use this really complex lateral line system to feel their way around. So they can feel their prey coming, they can feel predators coming after them, and they can even sense reflections of waves off the walls of the cave to help them avoid bumping into things. So if they're swimming towards something, they'll feel the waves coming back and they'll be like, okay, I'm getting a little close to the wall here. Let me back off a little bit. So this is something amazing these fish can do, and it's because they have this sort of overdeveloped lateral line system. So the fish that I study in my lab are called zebrafish, and this is a zebrafish right here. So you can see this fish is sort of more elongated, and they're actually quite small, so they only get about an inch long. That's what they look like from the top. So you can rotate them around. You can get them on your phone if you want using the QR code. Zebrafish are native to India, Bangladesh, and Pakistan, and they're a very commonly used model system. So what is a model system? A model system is an organism, a species that we can study to learn about things in other species. They serve as a model for certain things. So the zebrafish, actually has a lot of similarities to humans. You wouldn't think it looking at it, right? But zebrafish have a high genetic similarity to humans. We share about 70% of our genes. Just check the time. We share about 70% of our genes with fish, believe it or not. They're vertebrates. They have a backbone just like we do. They have all the same internal organs for the most part. Some differences. They have gills, we have lungs, but most things are pretty similar. They have a liver, they have a heart, etc. And so we can study things in these fish that we can't study in a human being, or even, let's say, in a mouse. So a lot of people use mice to study how things work in humans, but mice only produce a few offspring. These fish produce hundreds of offspring when they breed. And not only that, but you can study the entire development of the embryo from the time it gets fertilized. It's outside the mother, so you can study it really easily. And they're great for putting under the microscope, which is going to lead me to my next slide, which is this. So this is a young zebrafish, about 3 days old. And this fish was alive when I put it under the microscope. I captured this image myself. We immobilize and anesthetize the fish so they don't feel any harm or anything like that. They're not harmed in any way. They're just basically kept still. And then we can put them under a fancy microscope called a confocal microscope. And what that microscope allows us to do is to make a three-dimensional image of the fish. So what you're looking at is actually basically half of a zebrafish larva. It's missing the front of the head and the back of the tail. That's only because the field of view of the microscope was limited. So in other words, we couldn't capture the whole thing under the microscope, but the head is actually there and the tail is actually there in that fish. It just looks like it's cut off. And what I'm trying to show you is this fish has been genetically modified so that some of its cells express or produce a green fluorescent protein. So all these green things, all the green that you see is the lateral line system of the fish, which is producing this green fluorescent protein so we can see it against the background. So if we zoom way in, and I can actually center this and zoom in, so each of these little circles here is a little sensory organ. It's a little organ that contains a specialized kind of cell that lets the fish feel movement in the water. And these things are right on the surface of the body. So I'm going to rotate this, and you'll see they're kind of sticking right out of the side of the fish. I mean, it's a little hard to see from this angle, but there we go. So they're right on the surface of the fish, okay? And so that allows the fish to feel movement in the water, even things moving at a distance. If we zoomed in on one of these sensory organs, and the sensory organ is called a neuromast. So it's called a neuromast. It's a neural extension. It's basically an extension of the nervous system. If we look at one of these, we can see within it at a higher magnification the individual cells that are responsible for sensing movement in the water. And these are called hair cells. They're not like the hairs that grow on our heads. They're a totally different kind of cell. Hairs that grow on your head, or not my head, but somebody else's head, maybe your head at home. Those hairs are created by something called a hair follicle, which is like a little tiny organ, actually, that builds a hair. These hair cells are individual cells that have little tufts of sort of fibers coming out the top. And that's known as the hair bundle. It's like a little cluster of extensions from the cell. And if I rotate this, you can actually see those extensions sticking up. So these are the cells, these roundish things here, and I can rotate it this way so you can see a number of different cells in this cluster. And these are the hair bundles sticking up out the top of the hair cells. And these hair bundles are what detect movement in the water. So they stick out into the water. They're covered by a little bit of like a gelatin, which is called the cupula. So there's this cupula gelatin covering them to protect them. But they basically stick right out into the water. And when water waves hit them, they cause these cells to send a signal to the brain of the fish saying that something is coming. And so actually at the bottom of these cells, you can see these little pink sort of extensions at the bottom down here. I don't know if you can see those, but those are where these hair cells are contacting neurons. So neurons are brain cells. They're cells that are part of the nervous system. And in these fish, each hair cell has its own neurons connected up to it. And those send a signal to the brain when the hair cell gets sort of activated or stimulated. Now, how does the actual hair cell get stimulated? What does a hair cell really look like in greater detail? Because here you can see that they're sort of like, bulbous, and they have a little thing coming out the top. But if we were to look at a model of one of these, it would look something like this. So this is a hair cell. up here at what we call the apex or the top of the cell is the hair bundle. And you see it's got all these things sticking up out of it. Those are called stereocilia. And the stereocilia are what actually sense the movement in the water. And then this is the cell body. So most of the cell here, right? Here's the nucleus, like most cells, it has a nucleus, right? And then down here at the bottom, this is what's called the synapse. And a synapse is where a cell and two cells communicate to each other. For example, in this case, a hair cell communicating with a neuron, which are these yellow things. Okay. So this hair cell, let's see if I can rotate it. Ooh, that's not the way I wanted to rotate it. There we go. If we look at it from the back and we get rid of all the detail, this hair cell has all these neurons connected up to it, and it can send a signal to the brain when the hair bundle gets deflected, gets pushed upon. Okay. And I'm happy to answer any questions about more detail about how that actually works. So why are we interested in this? I mean, it's really cool, right? It's interesting how the fish senses things moving in the water. These cells are really interesting. But one of the things that makes us excited about these cells is in fish, when these cells die, the fish can replace them. They actually regrow or regenerate. So fish replace these hair cells throughout their lives. And that's probably important because those cells are kind of sticking out into the water. So you can imagine if any toxin gets into the water, if they brush up against something, they're going to lose some hair cells. So they have to be able to replace them. But what's really cool is that inside our ears, we have the exact same kind of cells, also called hair cells. So this is a model of the human cochlea. The cochlea is the inner ear, the part that does the hearing, basically responsible for the sense of hearing. And this is a section, basically a slice, through a long tube, which inside your ear is actually coiled. It looks like a snail shell. If you cut one little slice through that tube, you would see this, and it comes out looking like 3 chambers. And in the middle chamber, you have this structure here, which is called the organ of Corti. And if we zoom way in on this, and I can actually rotate it a little bit, so what you see here, these blue things are hair cells. And those hair cells, I can actually show you an even better view of them. I can make them kind of pop out here so you get a good look at them. These are the hair cells, and they form rows within the cochlea all along the length of the inside of the cochlea. I'll just let it keep playing for a moment. So those hair cells are responsible for our sense of hearing, especially these ones. These are called the inner hair cells. And what happens is when sound waves travel through your ear, they move this whole structure up and down, up and down. And when that happens, this membrane, this piece of tissue, pushes on the tops of the hair bundles, and that causes them to send a signal to your brain saying that you're hearing a specific frequency. So if we can understand how the fish replace those cells throughout their lives, which they can do naturally, but mammals cannot, we can't replace those cells normally. If we can figure out how the fish is doing it, because the fish has so much similarity to humans genetically and otherwise, maybe we can learn how to replace those lost hair cells in the human ear and actually restore hearing to those who have lost their hearing. So that's one of the things we're studying in my lab is how can we replace or produce more hair cells in order to eventually treat hearing loss. So that is, I think, pretty much all I have for today. I'm more than happy to answer any questions you may have. Looks like we're just about at time. So yeah, I don't know if I should consider, I'll continue sharing my screen in case anybody wants me to go back to any slides or anything like that. Yeah.
Sure. Thanks a lot, Dr. Steiner. Give a virtual round of applause. That was great. And so we'd love to have any questions. You can just shout them out, unmute yourself, or you could put it into chat. We'll do about 10 minutes of questions.
Excuse me. I don't know if this is true in fish, but maybe I don't know if this is true. I think in sharks, aren't they also able to sense like electricity or something with the lateral line?
They do sense electricity, but they actually use, that's a great question. They use a different kind of sensory organ called the ampullae of Lorenzini. Um, but they are related. They actually develop from the same kind of initial organ, but they differentiate, they change, they turn into, they produce different kind of cells. Um, so those cells do detect exactly electromagnetic energy. They can actually detect, like, the muscles moving inside a fish nearby, and they can go after it. Um, but no, it's actually not the lateral line exactly. It's a slightly different, like, modified system. But no, great question. Yeah, sharks can do that. Um, and certain other fish can do that as well.
Dr. Steiner, we have another question in the chat. They thought that
Okay.
the eardrum is responsible for hearing. Does it also help?
Yeah, so that's a great question. So the eardrum is part of it's sort of the window between the outer ear and the middle ear. I don't think I have a figure here that will show you exactly what I'm talking about, but basically sound waves travel into your ear, the outer part of your ear, which is called the pinna, or there are other names for it. Um, and that sound travels and hits the eardrum. When it hits the eardrum vibrates. Okay. That vibration actually drives the movement of three tiny little bones that are in your middle ear, sort of between the outer ear and the inner ear. There's this middle ear region. It's got three little bones in it called the malleus, the incus, and the stapes. And when those bones move, they basically cause a wave to travel inside the inner ear. So those bones translate the movement of the eardrum into a wave that travels inside the inner ear in the cochlea, and that's where the hearing actually takes place. So the eardrum is necessary for hearing, um, because it's that's where the sound waves initially hit, basically. But where the sound is detected is way further inside the ear. The cochlea is actually embedded in the bone of your skull. It's like way inside, inside your head. And that's where the sound is actually detected.
Thank you. We have another question. Fish have ears as well as humans. So do the fish have those hairs in their ears?
They do, yes, exactly. Fish do have ears, and fish can hear. They have a couple of different sort of ways of hearing things. They can sense movement, vibration almost using their swim bladder like a eardrum. But they also can sense vibrations other ways. And yes, they have hair cells in their inner ears as well. And just like the hair cells in the lateral line, the hair cells in the fish's ear can regenerate. Now, in my lab, we study the hair cells in the lateral line because they're really right on the surface of the fish's body. So it makes it really easy to look at them under the microscope. Um, because the fish is transparent, but if you go deeper into the fish, it's harder to see things. So, so we're able to see the cells really easily in the lateral line. And the other good thing about that is if we put, which we do sometimes, we put a chemical in the water that will temporarily kill the hair cells on the fish, but they regrow within about 3 days and the fish is completely fine. That happens with the lateral line hair cells. The inner ear hair cells are sort of harder to get to. So the chemical doesn't get there as easily. Um, so there are a number of reasons why we study the lateral line system in my lab. But yes, there are definitely people studying how hair cells regenerate in the fish ear also. They are there too.
Okay, we have a couple more questions. So how do the hairs feel through that gelatin thing?
Oh, that's a good question too. Yeah, these are great questions. So the gelatin thing, the whole gelatin thing called the cupula gets pushed, basically. And when it gets pushed, all the hair bundles that are stuck inside it. Um, so let me sort of go back to this slide. This might be helpful. So imagine here, whoop. too far. So there's a cupula covering this whole thing, okay? When that gets pushed, all the hair bundles simultaneously are pushed one way, okay? So that will stimulate the hair cells within that neuromast to send a signal to the brain through the synapses. So basically that cupula almost like synchronizes the hair cells, because if you push on the cupula, every hair bundle that's inside that cupula is going to get triggered. So that's how it works. It's flexible. It's basically like it's like gelatin, right? It like wiggles. So when sound waves hit it, it kind of wiggles back and forth, and that's what triggers the hair cells to fire.
Okay, you have a couple more really interesting questions.
Okay, cool.
So is the fish using the wave movement against the lateral line organs to create a mental map of what's around them in the ocean, the same way that we can tell where things are by seeing and hearing around them?
All right, somebody knows a little too much here. But yes, actually. Um, so when Dr. Fisher and I were in the same lab, when we did our postdoctoral work, and there's somebody else working there at the same time as we were, who was essentially studying exactly that. He was studying a different kind of fish, Aplochilus lineatus, doesn't matter what it is, but basically it's a surface dwelling fish. But it's right at the surface, and it has neuromasts on top of its head, and it can sense wave movements. Not only does it feel the waves coming in from a prey item that's nearby, but it can feel just by the waves the sort of the wavelength of the waves that are hitting its head. It can tell how far away the prey item is, because the waves stretch out as they travel. So wider waves mean the organism is further away. What it's trying to eat is further away. And they actually sort of integrate information from multiple neuromasts. So they can tell, like, the waves are hitting this neuromast and then that neuromast. The thing must be over there, right? So the thing must be in that direction. I don't know if you can see my hands motioning. I'm using my hands to motion as if everybody can see them, but they can tell directionality based on the order in which the sound wave or the waves hit the neuromasts. So yes, they basically are mapping their environment and figuring out exactly where things are. Those fish can, if you drop a bug in the water in complete darkness, they go directly to it with no, even if you take, it turns out if you make them blind, which you can do, they still do it. They still go directly to their food source.
Okay, so we have some practical questions here.
Okay.
So they were told not to tap on a fish tank because it can affect a fish's hearing. Can the constant tapping on a fish's tank permanently damage a fish's hearing?
Permanently damage, probably not. But can it cause undue stress on the fish? Yes. We actually use this as a test sometimes with certain mutant lines. So we have fish that have mutations that have an inactive lateral line. And if you tap on the dish, they basically don't respond. They just sit there. And then we have other fish that have a normal lateral line. If you tap on the dish, they're in like a little petri dish. You tap on the dish, they swim around all over the place. And basically, when you're tapping on the tank, that's what you're doing. You're stimulating their lateral line, and they think something's happening and they get stressed out. So, will it permanently deafen them? Almost certainly not. Will it unduly stress out the fish, make them concerned that something's going on in the water, there's a predator coming or something? That might happen. So that's really why you shouldn't tap on the glass.
Okay. Are there any other questions? I don't see any more in the chat. So if they're not, we'll thank Dr. Steiner once again, virtual clapping. And we'll send a follow-up message. You'll have a recording of not just a recording of this session, but we'll also give you access to all the slides and the interactive versions of everything. So you can have Dr. Steiner's guidance as well as the visuals to play with. So anyway, thank you all for attending. And on Tuesday at 8 p.m. Eastern, we'll have another session. We'll actually explore, with the guidance of a dentist, the inner workings of teeth beyond what you can normally see. It's it's a different view, and you might have more of an appreciation of exactly what's going on in your mouth. So thank you all once again, and I'll talk to you later Tuesday. Bye.
Thank you.